Hello, my name is Sae Komatsu, and I'm going to be a junior this fall at the Overlake School in Redmond. I will be talking about induced pluripotent stem cell-derived oligodendrocyte progenitor cells in secondary progressive multiple sclerosis, and I will be reporting on their survival rates and functional outcomes. So first, what are iPSCs and OPCs? So iPSC is skin or blood cells, also called fibroblasts, that are reprogrammed into an embryonic state to develop into any type of human cell. And then OPCs are multipotent cells that differentiate into mature oligodendrocytes, or OLs, to help maintain brain health and myelination. In demyelinating diseases like multiple sclerosis, many OLs become dysfunctional by getting damaged or destroyed. What is MS? So MS is any condition that causes, like a demyelinating disease is any condition that causes damage to the protective covering that surrounds nerve fibers, characterized by inflammation, loss of myelin, axonal and neuronal degeneration, and progressive brain atrophy. So first, MS arises from an autoimmune response where the body's immune system mistakenly attacks myelin. And then demyelinated axons are unable to conduct electrical impulses efficiently, leading to impaired nerve signals. And then lastly, this neurodegenerative process contributes to the accumulation of permanent neurological deficits seen in progressive forms of MS. Many anti-inflammatory drugs can delay or even eliminate relapses in RRMS, which is how MS initially manifests itself in most patients, but are still unable to stop the transition to the secondary progressive phase, which typically develops at an advanced age. In the MRI brain scans, you can see the lesions of SPMS patients compared to control groups. And then some symptoms of MS include sensory disturbances such as numbness or tingling, motor dysfunction, loss of balance, etc. The objectives of my study was to evaluate the survival rates and functionality of iPSC-derived OPCs in vitro, and then to assess the efficacy in promoting remyelination and disease stability of non-human models of progressive MS in vivo. And then these are just some of the definitions, but in vitro just means research done in a laboratory dish or test tube, and then in vivo is research done on a living organism. My method, so I included 5 studies and I did a comprehensive literature review, and then my search strategy included including keywords such as SPMS, OPCS, and MS. So this is the first study that I'll be mainly basing or focusing around. It includes 1 in vitro and 3 in vivo models for MS. So first, skin cells were collected from 4 healthy individuals to create iPSCs. And then the differentiation. So they reported that the efficiency of differentiation was 45% to 78% in different lines. To verify proper functionality, myelin formation in co-cultures of iPSC-derived OPCs with rat dorsal root ganglion neurons showed a survival rate much higher than observed in monocultures lacking DRG neurons. High magnification images such as this one reveals extensive myelination around DRG axons. After seeing the success for in vitro models, mice were induced with models of MS, with the first one being Cooper's own. The image here shows a greater formation of demyelinated lesions in white of Cooper's zone treated mice with OPCs. And then EAE was injected in the second model of mice, where OPC-treated mice showed a significant reduction in cell infiltration and demyelination compared to control mice, which is on top, and then OPC is on the bottom in the image. And then here, this graph shows in number of weeks that OPC-treated mice had a significantly lower EAE score than compared to their control counterparts, where a greater clinical score represents more signs and symptoms of neurodegeneration. These results show that post-injection of OPCs at 79 days after EAE induction slowed neurodegeneration, as shown in the graphs here. And then these images show OPCs in red interacting with axons in green, or the myelin basic protein. And then immunostaining reveals immature OPCs pointed by the arrows among the implanted cells differentiating towards astrocytes. And then finally, transmission electron microscopy reveals green OPCs wrapping extensions around axons in the lesions, forming thin myelin sheets. For the purpose of time, I am going to be skipping over studies 2 and 3. So study 4, part of the aims of this study was to ask whether iPSC-derived OPCs were able to develop into functional OLs and were able to form proper myelin around nude axons. To that purpose, researchers co-cultured the iPS-derived OPCs with a pure 2-week-old culture of DRG neurons. 2 weeks after they observed extensive myelin formation by the iPS-derived oligodendrocytes. Control DRG cultures, on the other hand, did not show any signs of myelination activity or myelin basic protein staining. And then, just as a reference, DRGs are right there in the spinal cord. So these results. So after 1 week, iPS-derived OPCs developed into myelin basic protein positive OLs that line their extensions along the neurofilament stained DRG axons. And then in the second week, OPCs start to myelinate the DRG axons in the environment. And then OPCs start to produce perinodin in red that at this stage is still being distributed over the extensions running along the axons. And then finally, after 2 weeks, long stretches of DRG axons have been myelinated by the co-cultured OPCs. So the last study focused on stem cell treatments on congenital hypomyelination using models in shiver mice. Although the study does not specifically target SPMS, it is similar to SPMS in the sense that it is a neurological condition characterized by insufficient formation of myelin, and it has similar symptoms. So essentially, this study shows that by 3 months after iPSC transplants, the white matter of recipients of OPC grafts manifested astrocystic scaffolds harboring densely engrafted myelogenic OLs, in each case yielding substantially reconstructed and densely myelinating central white matter. So these images show on the right is, or on the left, on the left it shows the 13 weeks where iPSCs matured into myelin basic protein expressing oligodendrocyte in green through the subcortical white matter. And then 6 weeks later, you can see the myelinated axons in red by the OLs. And then finally, the question they are asking here is remyelination noted in transplanted shiver mice sufficient to ameliorate neurological deterioration and prolong the survival of shiver mice, which typically die at 20 weeks of age? So iPSC OPCs were transplanted to 22 neonatal homozygous shiver mice, and then injection of saline to 19 littermate controls were performed, and then predictably, the 19 unimplanted shiver controls died before 5 months of age, while the 19 of the 22 implanted mice with OPCs lived longer than the longest lived control mouse. And then the graph shows that here in age and days, and then the percent survival between iPSCs and saline injected mice. The dot map indicates the distribution of iPSC-derived donor cells in the mouse brain. So then there's a quick anatomy of the mouse. So you can see it's really focused on the cerebellum and the hippocampus, which is really important for motor functions, etc. And then, so lastly, conclusion. Although MVP-positive oligodendrocytes constituted of varying percentages of total cell population, ranging from 18% to 70% in differentiation procedures throughout the studies, iPSC-derived OPCs selectively migrated to MS-like lesions and are capable of initiating remyelination of denuded axons within the inflammatory conditions of these lesions. The application of OPCs is particularly relevant to MS patients in the secondary progressive stage, when endogenous OPCs are no longer able to remyelinate and rescue damaged axons. The limitations of this research. So first, the reliance on animal cells. So non-human animal models may not fully replicate the complexity of MS in human patients. And then second, the use of non-autologous cells in vivo, which is basically saying that it may result in immunal rejection in human trials if the stem cells are not derived from their own. And then lastly, the lack of results on cognitive function and other symptoms of MS in vivo. So SPMS affects motor functions as well as cognitive functions like memory, attention, problem solving. So not assessing cognitive outcomes may overlook crucial aspects of the disease and not give us the full potential benefits of the treatment. Some future directions. Firstly, to conduct clinical trials to evaluate the safety and efficacy of iPSC-derived OPCs in humans with SPMS. The current challenges that inhibit us from doing so is the risk of forming tumors or uncontrolled growth, immune rejection, which I just talked about, and then ethical concerns. So the use of stem cells, particularly from embryonic sources. And then secondly, to optimize the differentiation and administration of iPSC-derived OPCs to enhance their migration specifically toward, so number one, clinically silent regions with limited chemotactic factor release that attracts OPCs to those myelin regions. And then secondly, regions that are enclosed by atrocystic scars from previous relapses in MS, and then long-term safety and stability. So to perform longitudinal studies to provide insights into the durability of the remyelination effects, and then long-term. So these are my references, and then thank you.
Thanks again, Sae. Any questions from the audience, or I'll check online. I don't see any current questions, but I open up the floor.
Hi, my name's Zara Rashid. I was wondering since why you chose to um, conduct your research on MS, and since MS is such a big field, how you ultimately ended up in researching ISPC derived OPCs.
Um, well, coming into this program, I was already interested in researching about stem cells. And then since this place is like focused on like neural, like spine anatomy and stuff like that, I just thought of MS since it's a neurological uh disease. And then yeah, I kind of just researched a lot. It took a long time for me to get to the final stage. But yeah.
I think again you did a fabulous job tackling a challenging subject. It seems like MS is something that we discuss every year. Last year, one of our research interns also did a multiple sclerosis project. I think you were innovative in the approach, trying to take your interest in stem cells and seeing how it's applied in this particular field, and choosing one of the types of MS patients and looking at the not only in vitro studies but in vivo studies and how we can apply it to the human condition. We had Dr. Rapovich
Mhm.
come here. He's a neuroimmunologist at Swedish, and he gave you some further perspective and insight. And so, are you a believer of the regeneration of myelin or slowing down of myelin in these patients? What are your thoughts based off of the research you've done?
I think that as long as there are human trials and then they continue to inject the iPSC derived OPCs,
Hmm.
then even if there is neurodegeneration, continually and consistently injecting that would probably slow the disease down
Hmm.
at at least, yeah.
Excellent. Any other questions before we? Okay. Oh, I'll ask a Dr. Tan question. 5, 10 years from now, where do you see yourself?
Um, Somewhere in the medical field, I'm not quite sure yet.
Okay.
Maybe surgery, and maybe like OBGYN, something like that.
Excellent.
Yeah.
All right. Thanks for sharing.